Hi, my name is Dinkorma Ouologuem. I am from Mali, and I'm a postdoctoral fellow from the Belgium program. I'm based at the Malaria Research and Training Center in Mali at the University of Bamako. I first earned a PharmD degree at the School of Pharmacy of Mali, and I was working on the human factors that were implicated in the clearance of chloroquine-resistant Plasmodium falciparum parasites. And then I moved on to the University of Pennsylvania in the United States to get a PhD degree in cell biology. And from then I moved back to Mali, where I'm actually doing my postdoctoral training at the MRTC. So when I was at the University of Pennsylvania, my doctoral training was on studying one parasite that is closely related to malaria that we call Toxoplasma gondii, and it was about learning how those parasites multiply. Now at the MRTC, I hope at the end of this training to be able to focus my work on the cell biology of Plasmodium parasites. So the community has made a lot of progress in the fight against malaria, but we have still not been able to eradicate this disease. And it has been recognized that malaria transmission from the human host to the mosquitoes vector is essential if we want to eliminate malaria. And there is an increased need to develop new antimalarial drugs and also vaccines to reduce malaria transmission in endemic areas. So my project will focus on the transmission of the parasite from the human host to the mosquito vector. So the parasite is really smart, and he has found ways to have multiple forms within each one of the hosts that it usually infects. And there is one particular one that the mosquito will take during its blood meal that we call gametocytes. And so what I want to study is how the gametocytes move from the human host to the mosquito vectors, and how those gametocytes are able to produce what will be injected in another human being and continue the cycle of the parasite. I will use the bioinformatic tools and the next generation sequencing technologies to identify marker or signature that are associated with the ability of those forms of the parasites that are transmitted to the mosquitoes vectors. And if I am successful, the study of the Plasmodium falciparum vectors associated with malaria transmission from human to mosquitoes hosts in our natural environment will generate new markers and targets for antimalarial drug vaccine development.